Now I would like to call on stage Dr. Manjari Jonnalagadda. Uh, she is a molecular anthropologist who is interested in understanding the evolution of complex traits among Indian population using phenotype, genetics, and evolutionary perspectives. In her doctoral work, she has studied the genetic variations in pigmentation throughout various social groups in Western Maharashtra. Now, let's gear up to our next talk, and Manjari ma'am is already here. She'll delve us into the implications of research in skin pigmentation studies on human health.
Hello, am I audible? Okay. Um, Good morning, ladies and gentlemen. My name is Manjari. I'm a professor of anthropology at the Symbiosis School for Liberal Arts, and I'm delighted to be here in front of this august audience. I would like to thank Professor Chaube and Dr. Chandana ma'am for giving me this opportunity to present my work. My presentation is titled Fair and Lovely or Dark and Beautiful because there's really no difference and they both are variations that we see in humans. Exploring skin pigmentation diversity in India. So my presentation will start with a brief introduction to the biology of skin color, and I'll then move on to global patterns of skin pigmentation diversity before I come to the South Asian context, where my work lies. So skin color is a result of the melanin pigment, and it's synthesized in organelles called melanosomes, found in specialized cells that are melanocytes in the innermost layer of the epidermis. And the skin color of an individual is a result of many factors. One is the migration of these melanocytes to this layer, which happens early in embryonic development. It's also controlled by the melanogenesis pathway, which is catalyzed by the enzyme tyrosinase, giving rise to two forms of melanin, that is pheomelanin or eumelanin. Eumelanin is characterized, or it's the dark skin, the melanin that gives rise to dark skin, versus pheomelanin that gives rise to the lighter skin. So ultimately, population variation is a result of both these processes, but also the number and the size of melanosomes in the cell and the aggregation of them. Relithford and many other anthropologists, they studied pigmentation variation and they found contrasting patterns between pigmentation diversity and the diversity of other markers. So in contrast to genetic markers and craniometric markers, pigmentation is seen to vary greatly among populations as compared to within populations, leading them to suggest that there is an adaptive feature or an adaptive basis for this trait. Uh, and no surprises there. Pigmentation strongly correlates with UV intensities, and dark skin is predominant in regions where UV radiation is high, and it lightens as we move towards latitudes where UV radiation becomes lower. Powerful hypotheses have been put forth to explain the emergence of these patterns, and I'd like to draw your attention to one of them. That's the folate vitamin D hypothesis by Jablonski. So it says that early humans were subjected to two opposing pigmentation clines, or they evolved to opposing pigmentation clines in response to UV. The first one is the folate-driven melanization, which we see in regions where UV radiation is high, characterized by, or which is characteristic of Africans and South Asians, so on and so forth, where high melanin levels, they form a natural sunblock to UV radiation and they protect against folate degradation. Folate is an essential nutrient that is involved in spermatogenesis and other reproductive mechanisms. And this degradation would definitely impact the reproductive fitness of an organism or an individual. So we see higher melanin levels in these regions. As we move towards latitudes where UV radiation is low, the melanin would probably be counterproductive. It would be blocking the UV radiation, whatever little bit is available in the environment. And it would also compromise vitamin D synthesis, because vitamin D is another biomolecule or a vitamin hormone that is synthesized based on exposure to UV. So in higher latitudes, the loss of pigmentation would have been favored naturally, and as it allows for vitamin D biosynthesis. So these patterns, they demonstrate how the evolution of these these pigmentation profiles look like. Coming to the diversity of pigmentation in global populations, I'm going to just run through these slides. Africa is the birthplace of humanity. We have earliest evidences of hominins and modern humans there roughly 300,000 years ago. And this long antiquity and occupation means that they have adapted to these climates. So it also means that they have greater genetic diversity, linguistic diversity, and this diversity is also seen in their pigmentation profiles, not as much as we see in other regions within the same continent. But what I mean is that even within African populations, we do see diversity in skin color. So data from few populations shows that melanin levels range from 50 to 100 melanin units, and most of these the genes that contribute to these patterns, they have been under selection for they have been under selection for these trait for in these populations, because they allow for adaptation, and they allow these populations to survive by repairing DNA damage and also preventing folate degradation. So this is what we see in Africa. The one gene that that's important here is the MFSD12 gene, among others, and dating of this variant shows that it emerged more than 500,000 years ago, preceding the origin of modern humans, indicating deep adaptive signatures. With the Out of Africa migration, we see that modern humans colonized different regions of the world, and Europe is characterized by low UV radiations, colder climates, and these populations are known to have lighter skin. Key genes that contribute to this phenotype in these populations are the genes involved in the potassium-dependent sodium-calcium exchanger family, the genes involved in the tyrosinase family, so on and so forth. And they are the ones that are involved in this phenotype here. What's interesting here is that this light skin probably didn't emerge immediately after the Out of Africa migration. It happened much later, because ancient DNA shows that early hunter-gatherers, European hunter-gatherers, had darker skin and blue eyes. So the introduction of this light skin allele probably happened with the migration of the Anatolian farmers, from West Eurasia. The emergence of, so one allele among, or one gene key among these is the SLC24A5 gene. It emerged somewhere around 20,000 years ago, but is fixed in this population, and it's dated to being fixed around 5,000 years ago. Coming to East Asia is also known to have low UV radiation, and these populations are also characterized by light skin. So uh, the genes, however, what's interesting in East Asia is that the genes contributing to light skin in this region are not the same that are influencing pigmentation in as compared to Europeans, suggesting a convergent evolution for this trait in this region. And key among these genes is the OCA2, that is the oculocutaneous 2 marker. The timing of this variant is dated to 25 to 30,000 years ago. It predates the variant that is seen in Europe, again highlighting the convergent nature of this trait in this region. We also see local patterns of local adaptation. For example, Tibetan populations live at high altitudes. They're known to survive at high altitudes. And the genes like the GNPAT are associated with facultative pigmentation. That's their tanning response to this high UV radiation. So these patterns clearly show that these populations have adapted to local UV regimes, and their pigmentation profiles have evolved such. Coming to South Asia, so I started my PhD work trying to understand whether pigmentation in India is more or less uniform or there is variation. There wasn't much data when I started my work, and of course, great work by Chandana ma'am also helped me to refine my hypotheses. So what we know about skin pigmentation in India, so before I go to what we see, the hypothesis here was that there should be some clinal variation as we move from north to south, maybe coinciding with UV radiation patterns like we see elsewhere globally. However, what we notice is that there isn't really any kind of patterning of pigmentation based on UV radiation. This is a map of many populations, of which the first six communities or populations in blue is what my data is. So from West Maharashtra. But when we compare it with other populations in India, you really don't see any kind of a clinal increase as we move from north to south. So these populations are arranged from central India to South India. Expecting an increase in pigmentation, we don't see that. Okay, so I'll come to what we see regional variation in pigmentation. Populations that are geographically contiguous are also showing high pigmentation diversity. And this is seen in West Maharashtra, the box that's labeled in red, as well as in South India. So, we have different populations sampled from South India, and we do see this kind of variation that exists in that region as well. Similar kind of patterns are observed in UP and Bihar. Fantastic work by Mishra et al. So this is their data set where they've mapped a whole lot of communities in these two states. And you again see a lot of distribution of melanin levels. What's key in these, in these density plots, so what can we take away from this is that firstly, pigmentation is patterned based on social groups. So if you categorize all these populations into the different social hierarchies that we see in India, where endogamous populations here occupying different sections of the society, so we have a social stratification. We notice that populations that are traditionally assigned to be upper castes, they have lighter pigmentation or lighter melanin or lower melanin levels as compared to populations that are traditionally assigned to be lower castes or tribal groups. And we also notice greater dispersion in their melanin levels as seen in those density plots. So clearly this is not a very strong UV induced pattern. We also wanted to look at what is the genetic basis for this diversity, and the first study that highlighted the GWAS in South Asia was done by Stokowski et al. They identified 3 genes and 3 loci that were significantly associated with skin pigmentation. We replicated that work in our Maharashtra data set. We find moderate to low association for these 3 genes. The key one is the first one, the SLC24A5 gene, and allelic distribution of this marker across India again highlights this lack of a clinal variation. So if you see the A allele is the allele that is associated with light skin, it's the derived allele. We see it being all over India. So there's high frequencies of the light skin in the north as well as in the south as well as in the west. So really there's no patterning as per UV radiation. If you look at the box plots, it's clear that individuals who are homozygous for the derived allele show lower melanin levels as compared to individuals who are homozygous for the G allele that contributes to dark skin. The box plot on the left is data from Maharashtra as compared to the box plot on the right, which is data from UP and Bihar. What's interesting is that from these two regions, we notice that individuals with the same genotypic background may show different melanin levels. So the melanin levels could vary slightly even if you have the same genetic genotypes. And that's something that's interesting. It highlights the polygenic nature of this trait. That means we really need to look for more variants that are contributing to this phenotype. In 2019, I had the good fortune and also the opportunity to do a GWAS study with the help of Dr. Para on looking at variants that contribute to pigmentation in West Maharashtra. Again, the genomic region in chromosome 15 that harbors the SLC24A5 genes, again showing the strongest signal for this phenotype. And we really didn't find any other genome-wide signals. So that's something that we could replicate based on what Stokowski and a lot of other people have identified. Another marker was identified by Mishra et al. That is again within the same gene. It's the RS2470102 allele. If you look at the distribution of this, the melanin levels across these three genotypes, they mirror what is seen in the SLC24A5, the first gene, the first variant. So clearly, this is an allele that is important and it's contributing to light skin in India. We also looked at what the distribution was of the other two genes, that is the SLC45A2 and the tyrosinase gene. And before I go forward, importantly about this gene, the variant has been dated to emerge roughly 28,000 years ago in the Levant region, and it probably entered into India through West Eurasian migration by populations carrying higher ANI ancestry. And so that's where probably the emergence of this trait in India and this patterning is based on. So it probably has a lot to do with ancestry as compared to a natural selection based pattern. These two genes, we found, like I said, very low association. We really couldn't replicate these signals in these populations. This is a map that's seen in India for these two SNPs, and whatever comparative data was there, we kind of pooled all of it together. The pie charts that are in star are from my data set from Maharashtra. We really don't see great differentiation based on these two alleles, so we really don't think that there's much happening here. It's not contributing to pigmentation greatly in this region. Okay. So this is the story of these three alleles that were identified in Europe, and we try to see what's happening in India. In the 90s, Harding et al. And other scholars, they highlighted the selective constraints on MC1R and highlighted MC1R as a candidate gene for pigmentation, and they showed differential patterns of selection on MC1R in Europeans versus Africans. And we tried to see whether MC1R may be influencing pigmentation in this region, and to see whether dark skin populations are demonstrating some kind of selective constraint. We did not see any such kind of signal. MC1R is not under selective constraint in West Maharashtra, and it is unlikely to influence pigmentation. So that rules out another gene for us. We also undertook a scan of selection signals in the Gujarati Indians and the Indian Telugus from 1000 genome database to try and see if we could identify more pigmentation genes. We identified 27 of these genes which are known to influence pigmentation based on the pigmentation database. But out of these, 8 have been previously associated in prior studies, and key among this, which I think deserves more attention, are genes like PDPK1, LYST, MLPH. These are melanosome transport genes, and PDPK1 is recently associated with dark skin in Africans. So these are the genes that overlap between GIH and ITU and which are unique to GIH and ITU. And as we can see, GIH is the only population out of these two that is showing the SLC24A5 gene. It's not seen in South Indians. And this is known because, like previous work by Chandana ma'am has shown that this gene is under positive selection in North Indians and not in South Indians. So we went back to see what is, if there's some kind of a demographic basis to explain these patterns. Why do we see, such kind of diversity and all? So we did an IBD analysis of our populations, and we clearly see clustering of the caste groups versus the tribal groups. Now, this doesn't directly inform pigmentation, but it can form a strong basis to explain patterns of diversity that we see. And these confirm known signals of population affinity between Bhils and tribal populations of that region as compared to caste groups. But what is interesting is the separation of caste groups versus the tribal groups. We also did another study where we looked at ancestry patterns, and this was something that came that someone suggested to me yesterday in my conversations, where we have done an ancestry mapping of our samples versus all other samples where we know ancestry estimates for, and we try to see what is the ancestry estimates for our populations and what are the corresponding melanin levels. It's clear that our tribal groups show higher ancient ancestral South Indian ancestry, the blue bar graphs there. But their corresponding melanin levels are not very high, so they may be marginally high. We've not done very strong correlation. We have not done correlation studies, so this is just an indicative pattern as of now. But maybe there's something there. And we've seen what's happening with the caste populations. They show much lower AASI ancestry, and they also show lower melanin levels. So, what do I want to say with all of this? Firstly, pigmentation is very diverse in India. It's regional as well as within geographically contiguous populations. It's patterned across social groups and social hierarchy, and that reflects our population stratification, our population structure. We are endogamous groups, we marry within ourselves. Okay. It also does not correlate with UV intensities as we would think it does. The SLC24A5 gene, A allele, and the other SNP contributes to light skin in this region. We really don't know much about what's happening with dark skin, and it may have strong correlations with ancestry and ancient migrations into this region. So, coming to the future implications of this work, we really think that we should be looking for adaptive signatures of dark skin in Indian populations. It would be very interesting to integrate genomic data with evidences from paleoclimate, because there are very interesting studies on paleoclimatic research that are trying to model what paleoclimatic environments look like, and maybe those studies can give us good indications to what kind of genomic segments we should focus on. We also don't know anything about the tanning potential of Indians. Maybe this is a phenotype that has really not changed since we migrated from Africa to Asia. But how do we cope with local UV intensities is by increasing our tanning potential, right? So maybe constitutive pigmentation has not changed much, but we have improved our tanning potential, and that's how we have managed to kind of adapt to these climates. So that's an area of future research. We also need to understand the diversity in other pigmentary traits that could have both pleiotropic and epistatic effects. So we also need to understand the influence of social and cultural practices on skin pigmentation. It's strongly patterned as per social hierarchy and social groups. It would be very interesting to extrapolate this work to understand local adaptations to environment, to diet and diseases. Obviously, there is independent inquiry into these domains, but I think looking at it from a holistic and an integrative perspective may give us better ideas as to how what's happening. Implications for human health involve understanding pigmentary disorders. If we are able to delineate patterns of normal pigmentation and the processes that contribute to them, we should be able to, or it will definitely help to talk about how pigmentary disorders work. These are 3 genes that we have identified in our selection scans in Gujarati Indians and Indian Telugus. And these are genes involved in melanosome transport, so maybe they could be investigative. They could be markers that we investigate in future to identify and understand pigmentary disorders. We also need to understand the link between skin pigmentation and vitamin D deficiencies. This is it's in fact becoming a public health challenge today in India. Living in a warm climate with ample sunlight, a lot of us are vitamin D deficient, and that deficiency is seen across cohorts. So we really don't know what's happening in India. So these are possible and probable areas of future research. So with that, I come to the end of my presentation. I have thanks to give to the funding agencies. This work was supported by SIR through an earlier career research grant to me, and also through the Women Scientist grant that was awarded for my PhD work. I also have to thank my collaborators, Dr. Esteban Para, Heather Norton, Richa Ashma is my PhD guide. The late Dr. Shauna Kulkarni is also my co-guide. He's my guide. Richa Ashma is my co-guide. This is fairly emotional for me. This because we lost him to COVID, and he has not seen the completion of this work. And Dr. Shantanu Ozarkar, who's been a long-time collaborator and my partner. So thank you so much.
Yes, Satish. One or two. Yeah, it's wonderful, wonderful work. Just an observation from
Absolutely, thank you.
previous talk and this talk.
Yeah.
Genotyping is not an issue. Only is the money and the sequence. The issue is how you can refine your phenotype.
Yeah.
That's one. The second issue is when we talk of any statement about say Indian population, the only thing that we say that any statement that is true in India is that no statement is true in India. The moment you say South India,
yeah,
I mean, Haryana is staying for 30 years in South India. My children don't speak Telugu, they don't speak Haryana. Somebody comes there and they take the sample, they will be classified as South India. So this is where my message is we need to be very, very careful while doing these any genetics of the human beings. From violet top, the kind of phenotype on your crop define the population. That's very, very critical, and particularly for the youngsters who are initiating themselves, go back, read Mendel's paper English translation. They are famous for what he did, but you will find they should be famous for how to do an experiment. Know your population, clean your population, and then do something. That is where the trouble is. So what was that? Thank you very much.
Thank you so much for your comment. Yes, sir, absolutely. I totally accept.
Yes, the trash and another one.
Wonderful talk, Manjari. Wonderful talk. I just thought,
Thank you.
There could, there could be, a kind of a selective pressure, that might be sitting, largely associated with healthy, linkage disequilibrium.
Sure.
I mean, I couldn't see, a large amount of GC and NKFR2 genes, where
I'm sorry, I can't hear you very
called
clearly.
GC and
Yeah.
NKFR2, which are well known for skin pigmentation and vitamin D. So these were relatively well known in a large amount of studies. And there could be a plausible reason betray that SLC 24A5 is largely co-expressed with GC. So GWAS couldn't probably give these kind of certain answers.
Yeah.
So if you can probably go deeper and go for a targeted genome approach across a diverse phenotype genotype correlation.
Yes.
And try to dig into that. And I'm sure that answering those linkage disequilibrium in lieu of the polygenic discourse can largely be, giving a large amount of answers.
Yes. Thank you so much for that suggestion.
Yes, yeah, I feel I'm audible from here. I'm audible from here to everybody. So it's a particular way that because you are from anthropology and I'm from anthropology. So there is a connection. So one thing it would have been better to refer a ecological route by Glauser's route, 1833. That you are talking about this latitude and the differences of the skin color. So for the purpose study, I feel that's a respect to Glauser and the ecological rule in 1833. And the next one is you are talking about the tanning, possibility of the tanning. So I guess this is the artificial skin color. You are going with the skin color, I mean the dimensions, I mean the quantitative characters of the skin color you are taking in spectrophotometer.
Yeah.
There is exposure. So if you can make a differentiation, I mean the difference between
we have not done that as yet,
So just
but yes, that's something that we are thinking about.
How this scanning, how much the population gets.
Yes, it's difficult to characterize tanning because you really can't quantify how much time people spend out in the sun. It again has a lot of social cultural dimensions.
Occupation is also very
Yes,
important.
but we are thinking about a model to develop that to and measure that. Thank you.
So a quick question. So in your data in the Tibeto-Brahmin population in one of the SLC,
Yeah.
I saw a very skewed value distribution, but you
Yeah.
didn't comment on it.
So yes, so that was comparative data. It's interesting. The SLC 24A5 has, if I just go back to that slide, it has higher frequencies of the ancestral G allele. So there, but we think it may be a reflection of their ancestry components. It may not be, because that's what we're seeing in the rest of the region as well. Populations that have higher ANI ancestry are showing lighter skin as compared to those regions. So I haven't looked at the ancestry components of those populations, but I think it may be a reflection of that.
Dr. Jonnalagadda, one issue, we have to see beyond.
Okay.
These genes are there only for skin color. That is maybe one problem. I see all these genes. Some of these genes are cell communication. So some things are the calcium, potassium channels.
Yes.
Very important genes in terms of other physiological processes.
Yes.
Okay, still maybe an indicator for you. In fact, skin as a resistance doctor, we have indications that skin patches can lead to some chronic conditions. Some of understanding is that.
Yeah.
But right, looking into those genes or GR genes, so many discoveries. Adam1 gene. It is a cell-cell communication, a very important gene for several other processes. So, part biology.
Yeah.
Doing this is fine,
Okay.
selection. But going to beyond, part biology, trying to understand why these genes are there. But, okay, you may be getting some indications for, ultimately lifestyle.
Okay.
Lifestyle diseases, vitamin D3, vitamin D deficiency. It's a big issue in human population. We all know that. These are also related. So look into part biology.
Thank you so much for your suggestion. I've written it down.
The next year. Yeah.
I have a